We'll now move on to our last talk in the session, Dr. Anita Sil from UC San Francisco on virulence of coccidioides.
Okay, great. Wow, it's very confusing. Jason's slides are still up here, but mine aren't there. Okay, thank you. This is a great meeting. I'm so grateful to be here with all of you and all of the people who are here virtually. I am going to talk about virulence gene discovery in coccidioides today. I think it's very straightforward to appreciate that to understand what the fungus is doing when it's causing a disease in the host, it's important to understand this developmental transition from the arthrochonidium to the spherules, which is critical for disease-related biology that occurs during infection. So I'm going to tell you today about some of our studies thinking about the regulation of this process, as well as the molecular transformation that accompanies this morphologic and developmental transition. Okay, so I wanted to give a shout out at the beginning to Donna Love from NIAID, whose vision I think really bolstered the development and the opportunity of these coccidioides collaborative research center grants. There are a number of these. There are a lot of folks who are in these different cocci centers who are here at the meeting, and it's a wonderful way for members of the community to bring our research efforts together and have a lot of collaborative discussions that I think will bolster the progress in this field. So our lab, along with a number of PIs, some of whom are shown on the slide, has one of these center grants called Molecules and Pathways at the Coccidioides Host Pathogen Interface. And we have 3 projects as part of this grant. The first project is based on identifying mechanisms of fungal virulence. The second is more focused on host response. And the third is looking at immune and metabolic correlates of coccidioides disease and all of its different outcomes, from disease that's resolved to disease that's disseminated. And what I'm going to focus on telling you about today are some of our efforts as part of this group to look at to identify potential fungal virulence factors. So as Bridget mentioned a few moments ago, I'm going to talk about looking at the transcriptional program of coccidioides development. And then I'll tell you a little bit at the end about some burgeoning initial studies looking at the interaction of coccidioides with innate immune cells. So really building on the work of many people in the field, a current adult infectious ID fellow in my lab, Christina Homer, developed some conditions to get reproducible and robust development of spherules from arthrochonidia in vitro. And you can see here, these are triplicate samples going through this developmental transition in a relatively high density time course from arthrochonidia to spherules and then to spherules releasing endospores. And Christina took these samples and subjected them to transcriptional profiling so that we could identify the transcripts that change in abundance over this developmental time course. And here you're looking at a heat map. And so for folks who aren't used to looking at these that often, every row on this heat map represents a different transcript, and each of the columns is a different experimental condition. So on the left, in those first 3 columns, you have the arthrochonidia, and then over time, just like that time course I showed you, there are triplicate samples for all of those time points. And yellow indicates an increased abundance of a particular transcript, and blue is a decreased abundance. And so you can see in that left-hand side, there's a pattern of transcript abundance that that the arthroconidia show, and then that changes dramatically as they develop into spherules. And so, a large fraction, over about a third of the transcripts in the genome are changing their expression pattern over this time course. And so one of the questions, so that's very interesting to us. There are a number of transcripts that are, more present in spherules than they are in other developmental stages. But how do you go from such a large data set to kind of interrogating, individual transcripts that might be important for disease? And so one way we're doing this is we're looking at the regulation of this program. And so I want to tell you a little bit about some work that we did in collaboration with Mark Orbach. And this is looking at a transcription factor in coccidioides that's important for this developmental transition into spherules. So transcription factors are proteins that regulate the expression of groups of other genes. And we had identified a transcription factor in a different thermally dimorphic fungal pathogen, histoplasma, that you heard a little bit about earlier today, called RIP1. And RIP1 is important for the transition of that fungus, histoplasma, from the soil form into the host form. And so Ali Mandel and Mark's lab deleted the RIP1 gene in coccidioides, and Sinem Behan, who was a postdoc at that time in my lab, got some RNA samples from Ali and looked at the transcriptional profile of the RIP1 mutant samples versus the wild-type samples under spherular conditions. And we found that RIP1 was important for some of that spherular transcriptional program that I just alluded to. Additionally, Ali and Mark showed that the RIP1 mutant cannot make spherules, and then in collaboration with Lisa Shubitz and John Galgiani showed that the RIP1 mutant is avirulent in the mouse model of infection. So all of this is telling us that this transcription factor is very important for driving the spherule program, part of the accompanying transcriptional program, and virulence in the animal model. So we regenerated or recreated the RIP1 mutant in our lab at UCSF, and Christina did, again, this similar high-density developmental time course, either with wild type, making spherules, or with the RIP1 mutant, which makes, sort of these aberrant morphologic forms. And she repeated her transcriptional profiling with these samples in parallel, wild type and mutant, and it generates a very complex data set. And today, for the interest of time, I'm not going to go into that in a lot of detail, but I want to give you an example of how this type of work helps us pick out potential virulence factors. And so to do that, I'm going to talk very briefly about a virulence factor that was identified previously in the literature. This is a protein many of you are familiar with, SAO-GP, which is spherical outer wall glycoprotein. It was identified by Chung-Yu Hung in Gary Cole's lab a number of years ago. And the protein is an immunodominant cell surface proline-rich antigen that's expressed only in spherules. And if you mutate SAO-GP, they showed that those mutants have attenuated virulence in a mouse model. So Christina in her transcriptional work could see, as recapitulate the work of others and see that the SAO-GP transcript, so the transcript that encodes that protein, is highly induced as cells make spherules. And interestingly, the expression of SAO-GP is dependent on that transcription factor, RIP1. So you can see in this wild type and RIP1 mutant time course, you see that the SAO-GP transcript is induced in wild type as the cells make spherules, and that expression drops a little bit as the cells move into the endospore form. It's not induced in the RIP1 mutant under spherulation conditions, so the expression is dependent on RIP1. And then, as expected, you don't see it under hyphal conditions at all. So how does this help us identify other genes that might be important for virulence? So what we can do, so it's a common principle in pathogens that virulence factors can show co-regulation transcriptionally. And so if we look in our data set at other genes that are co-regulated with SAU-GP, which is the top line of this heat map. The transcripts that fall into this category that are also dependent on RIP1 are very interesting candidates for new potential virulence factors. And I'll just point out that this cluster includes a urease transcript, and that urease gene was previously shown also to be important for virulence. So there's only a handful of known virulence factors in coccidioides, and this type of discovery work we hope will be really important for identifying new molecules and pathways that are important for virulence. So in the future, we're going to be taking these candidates and mutating them in coccidioides and looking at the effect in the mouse model of infection. Okay, so I'd like to tell you just a little bit at the end then about what we're trying to look at in terms of coccidioides and innate immune cells. So there's some information in the literature about macrophages and the interaction with coccidioides, but not a lot is known. And so we've decided to take just a model macrophage system, so starting out with murine, so mouse-derived, bone marrow-derived macrophages, and just look to see what happens when we infect with arthrochonidia. And I'm not, again, going to show you all the data, but we can see that those macrophages will take up arthrochonidia. And then you get the appearance of spherules, some of which, and you get the death of the macrophages. So I wanted to show you a couple of movies about how these macrophages stimulate the appearance of spherules. So if you take arthrochonidia and you incubate them under, in macrophage media, but without macrophages, what you see is the development of hyphae, so the same form that we would normally see in the soil. So these movies are taken, there's one image taken every hour for 3 days, and you can see that you get this robust mat of hyphae forming. In contrast, under the same conditions, if you provide macrophages, and those are the large cells that you're seeing here, what you see is that those arthrochonidia, instead of making hyphae, will transform into spherules, and actually quite large spherules. You can see the appearance of, especially in the right-hand side, some very, very big spherules here. And as I told you before, a moment ago, in the absence of those macrophages, if you look in the left-hand side here, you'll see one spherule appear, but the majority of the cells don't undergo this developmental program. So that suggests that there's something about the macrophages that's stimulating formation of the host form. And this harkens back to some observations that John Galgiani made years ago, seeing that, in the presence of mammalian immune cells, you see this, similar stimulus for the fungus to transform into the host form. Interestingly, it appears that you need contact to have this happen. So if you separate the macrophages and the fungi, you don't see formation of spherules under these conditions. And so we're very interested in working out what's happening at that host pathogen interface to figure out how stimulation of the spherule form occurs. Okay, so what I've told you is that there are some dramatic molecular shifts that occur during spherulation. And we know from work with Mark that there's a transcription factor, RIP1, that's required for that spherulation program. And I didn't show you these data, but RIP1 is actually important for parts of both the spherule and the hyphal transcriptional program in coccidioides. We're looking at spherule-specific transcripts to interrogate them for roles in virulence, and we're interested in understanding how it is that macrophages stimulate spherulation. So I want to thank the folks that have done the work. So in the highlighted individuals, so Christina Homer is a fantastic adult infectious disease fellow in the lab who's looking at the developmental process of spherulation. Jane Symington is a fantastic pediatric infectious disease fellow who's looking at the interaction between macrophages and coccidioides. Mark Voorhees is a bioinformaticist in the lab who is involved in all of our large data set analysis. And I want to thank the former members in the lab who really started the coccidioides work. I'm a histoplasma biologist for most of my work and have really enjoyed coming into the coccidioides field. Sinem, when she was a postdoc in my lab, did the work that I talked about with RIP1 in collaboration with Mark. And she has her own lab at JCVI and some affiliation with UCSD. And then 3 other folks, Jesse Euling, Bastian Junk, and Keith Walcott, were really instrumental in getting a lot of the coccidioides culturing and mutant generation in our lab at UCSF. And I want to thank especially Mark as well as Bridget, who's been an amazing collaborator and has really helped us get everything set up. Thanks.